Hello students, let us continue our discussion regarding chapter respiration in plants. In the previous class, we were giving much importance regarding the electron transport system, which is a part of the oxidative phosphorylation concept, which ultimately leads to production of the 38 ATPs by the oxidation of energy rich molecules like NADH plus H plus and FADH2. The process of formation of ATP by a chemiosmosis, it was explained by a scientist called Peter Mecher. How exactly these ATPs are produced, and for that we had gone for some of the assumptions. If you remember, assumptions we had taken, and what were the assumptions? Quickly tell me students. Assumptions were, the first assumption was glucose is the only one respiratory substrate used for respiration. Second, NADH plus H plus produced during the glycolysis enters the matrix of mitochondria for production of which is involved in oxidative phosphorylation, second one. The third point is the glycolysis, Krebs cycle, oxidative phosphorylation occurs in a sequence wise, third point over. And the important thing is none of the intermediates of the aerobic respiration, maybe in glycolysis, maybe in Krebs cycle, they do not escape the respiratory pathway. Clear? With these assumptions, we get 38 ATP. Clear, right friends? Okay, how the 38 ATPs, actually we had already gave the idea regarding that. So let me go for that now. Students, in the glycolysis, quickly tell me, in the glycolysis, how many ATPs are produced? How many NADH plus H plus produced? You should tell me. And how many FADH2 produced? These three aspects you have to tell me. Okay. In the glycolysis, and this is this ATP by substrate level phosphorylation. The next one in the glycolysis, then it is regarding the oxidative decarboxylation. The last one, the glycolysis oxidative decarboxylation. Next one is the Krebs cycle. Then it's regarding the it's fine. So let us go for totaling later. Glycolysis, how many ATP? Tell me. Net yield we should take it. We should take only net yield. So 2 ATP from the glycolysis. How many ATP during the oxidative decarboxylation? SLP not there. How many during Krebs cycle? 2 ATP. So that means totally there will be 4 ATP total, right here, totally there will be 4 ATP by the substrate level phosphorylation. Clear, right? Which do not require any chemiosmosis. Let us come to NADH plus H plus. How many NADH plus H plus here? 2 NADH plus H plus. In which step, please tell me? Oxidation step, when glyceraldehyde 3-phosphate becomes 1,3-bisphosphoglyceric acid. In the 6th step of glycolysis, 2 NADH plus H plus is formed. Okay. Oxidative decarboxylation, which step, tell me? When a pyruvic acid becomes acetyl-CoA. During that step, 2 NADH plus H plus is formed. 2, all right. Krebs cycle. 3 places. Can you tell me the places? Tell me fast. Yeah, when isocitrate becomes alpha ketoglutaric acid, when alpha ketoglutaric acid becomes succinyl-CoA, and when a malic acid becomes oxaloacetic acid. 3 places the NADH plus H plus formed. So write 3 here. 3 places. 3 places means 6 here. 6. Okay. Now all together tell me. Yes, 10 NADH plus H plus over. Okay. 10 what? 10 NADH plus H plus. So, I will write here 10 NADH plus H plus into 3 you make it, into 3. So, it gives 30 ATP for us, 30 ATP. Clear, right? Okay. 4 ATP already we have it here. How many FADH2 in the case of glycolysis? 0. How many in the case of oxidative decarbonation? 0. How many in Krebs cycle? 2. In which place, tell me? When a succinic acid becomes fumaric acid with the enzyme succinate dehydrogenase, those two FADH2 form. So, I will write here 2 FADH2 into 2. So, it becomes 4 ATP, 4 ATP. So, 4 ATP there, 30 ATP here, 4 ATP here, students. So, together how many ATP, please tell me? Together it is going to be total number of ATP. I will write here, it is going to be total ATP for one glucose molecule. For one glucose molecule during the aerobic respiration, during the aerobic respiration, tell me how much it is. It is going to be right here 38 ATP, 38 ATP. This has to be remembered. Please do not forget this. How many by substrate level phosphorylation? How many by the oxidative phosphorylation? That segregation also you get it here. These are by oxidative phosphorylation. This is by substrate level phosphorylation. This requires chemiosmosis. This requires chemiosmosis. This does not require chemiosmosis. All this has to be remembered, students. With that, I am erasing this respiratory balance sheet for one glucose molecule giving 38 ATP. So, 38 ATP can give 686 kilocalories of energy. Now, we will be heading for next aspect. The next aspect is going to be when we compare aerobic respiration with the anaerobic respiration, how they are different. The point has been given here. Just you have to listen to that and you have to take down in your book also. Comparison between fermentation and aerobic respiration. Let us see. First one, fermentation. It is a partial breakdown of the glucose molecule. Fermentation, is it a partial breakdown of glucose molecule? You should tell me the answer. Answer is yes. How? Tell me. Because in a fermentation, it may be either ethanol formation take place, C2H5OH ethanol formation, which has still a carbon-carbon bond here. So, it is a partial breakdown. It may be a acetic acid formation, not acetic acid, lactic acid, maybe acetic acid also, but here I am taking lactic acid. It may be a lactic acid formation which has 3 carbon, that means 2 bonds are still left out, 2 carbon, still 1 bond is left out. So, it is an incomplete or partial breakdown of the glucose, which one? Fermentation. Aerobic respiration, it is a complete breakdown of the glucose into CO2 and H2O. Complete breakdown of the glucose, where it has become CO2 and H2O in the Krebs cycle, where all the bonds are broken to become CO2 and H2O, correct. Net yield of ATP is 2 ATP in fermentation, which is formed during glycolysis, if you remember. And number of ATP here formed is 38 ATP, I will write once again here. Number of ATP is 38 ATP is formed during the aerobic respiration. Do not forget that 38 ATP formed during aerobic respiration. Then in the fermentation, what happens? sixth step of the glycolysis, NADH plus H plus was formed. That gets reoxidized during the fermentation. See, the NADH got oxidized to NAD plus rather slowly in the case of what, tell me, the fermentation. But here NADH is oxidized to NAD plus very vigorously during the oxidative phosphorylation. Clear, right? NADH plus gets oxidized in NAD plus during oxidative phosphorylation, which occurs vigorously compared to the fermentation. So ultimately, tell me, energetically, which is very useful process, it is the aerobic respiration, because it is going to give how many ATP, tell me? It is going to give the 38 ATP. But in contrast to that, the anaerobic respiration, if you take it, it is going to give less number of ATP. Students, I will be heading forward for next concept here. The concept which I am going to discuss will be called as respiration as an amphibolic pathway. How it is? Let us discuss now. See carefully now. So, I will be taking here the glucose. Glucose will undergo oxidation, and ultimately becomes here two chemicals. One is called as I will be writing the glyceraldehyde 3-phosphate here, the glyceraldehyde 3-phosphate is a one molecule. The another molecule here is going to be dihydroxy acetone phosphate, dihydroxyacetone phosphate here, DHAP. Here let me write here DHAP, dihydroxyacetone phosphate. It is a glycolysis, right, where glucose is utilized. Glucose is the carbohydrate, it is a carbohydrate. Okay. Now, you know that dihydroxyacetone phosphate undergo reversible reaction and becomes once again glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate is going to become 1,3-bisphosphoglyceric acid. 1,3-bisphosphoglyceric acid. How many molecules? Also you have idea, 2 molecules here, and during this time NAD H plus has been formed. Then the cycle continues and continues and continues. Okay. I have not maintained the steps specifically. We can maintain it. How many steps? Glucose to the here it is the 5th step actually. I can write here the 5th step, and this I am writing the 6th step, you remember. Now I am directly going to write the last 2 steps. So I will write here the chemical name is phosphoenol pyruvate, PEP, which is going to give the pyruvic acid. Clear, right? And pyruvic acid has how many carbon? Tell me. 3 carbon, that means 2 molecules. So which step I have written here? 10th step I have written here. 10th step I have written here. Clear, right, friends? Okay. Now, so this first, the glucose to pyruvic acid formation is a glycolysis, and carbohydrate is used for that. One segment over. Now, later the pyruvic acid will become which chemical? Tell me. Acetyl CoA. Acetyl coenzyme A. And acetyl coenzyme A will, let me write the acetyl coenzyme A like this. Yes, acetyl coenzyme A. The acetyl coenzyme A will enter the, tell me, enter the Krebs cycle. I will write here now, enter the Krebs cycle. In the Krebs cycle, what happens? Tell me, students. The Krebs cycle, which you have already seen there will be decarboxylation, and there will be dehydration also. So, there will be decarboxylation is occurring. So, we tell CO2 plus H2O, right, and H2O here. Yes, clear, right? It is regarding the aerobic respiration pathway. I written now here. You can see here carbohydrate ultimately becomes here CO2 plus H2O. Yes. Now, this is happening when a glucose is used as a respiratory substrate. But can anything else used as respiratory substrate? And that is possible. And what is that? See here. When a glucose concentration is going on becoming less in the body, clear, preferably in the case of us, when we go for fasting, the glucose concentration goes on becoming less. Glycogen also will be broken down by glycogen lysis with the help of a hormone called glucagon, and that is going to become glucose, and glucose will be broken down into all this pathway. Imagine that all the carbohydrates are utilized. And what is the next source? The next source can be fats, lipids. Lipids can be the next source. So, the fat word I am writing now, lipids can be next source here. So, I will write here fat. Fat will be, you know that fat is made up of the glycerol and a fatty acid. The fat is made up of glycerol and fatty acid. Clear, students? Now, what can happen is this glycerol can become glyceraldehyde 3-phosphate. Glycerol and fat means fat is made up of fatty acid and glycerol. Hope you remember in the chapter biomolecules also we studied a fat is a esterified molecule. Triglyceride we called it as. Simple lipid means triglyceride, where one glycerol molecule, 3 fatty acids we had seen. 1, 2, 3 fatty acid. This is a glycerol molecule. They are bonded by the ester bonds. Yeah. So, that means if I remove all the fatty acids, then fatty acid plus glycerol molecule left out, right? Same thing we are telling here. So, when the fat is broken into fatty acid and glycerol, that glycerol molecule can undergo phosphorylation to become glyceraldehyde 3-phosphate. After becoming glyceraldehyde 3-phosphate, it continues the respiratory pathway. That means fat can link into the respiratory pathway. Can a fat enter the respiratory pathway? Answer is yes. Through what? Through glyceraldehyde 3-phosphate, the glycerol molecule. Clear? Next one. Can a fatty acid enter the respiratory pathway? Answer is yes. The fatty acid can enter the respiratory pathway through the acetyl-CoA. Through the acetyl-CoA, fatty acid can enter the respiratory pathway. That means a fat can enter the respiratory pathway in 2 ways. One is glyceraldehyde 3-phosphate, second is fatty acid through the acetyl-CoA. I think this matter is clear for you. Where it occurs preferably, I told you it occurs in the case of us during fasting. Second, in the case of the animals living in the desert, where, like I can give example, kangaroo rat, camel, they have a stored fat in them. This stored fat is broken like this, and the molecules enter the respiratory pathway. That gives them energy, that even give them water, which is called as metabolic water. They use that metabolic water. Which metabolic water? The metabolic water which we had seen in the oxidative phosphorylation also. Remember that. Okay. So, that is regarding fatty acid. Next one, let us come here now. Okay, when a fat is also utilized now, fat is utilized. Is anything left out? Yes, protein is there, right? Protein. Protein is a chain of amino acid, right? A protein, a chain of amino acid bonded by the peptide bond. First, the enzymes called as proteases, protease type of enzyme. The protease type of enzymes will break the peptide bond and ultimate source will be amino acids. Amino acids will be formed. Clear, students? Okay. Can amino acid directly enter the respiratory pathway? Answer is no. Listen carefully. Now, first step has to occur. I am drawing here to give an idea to you. Alpha carbon, COOH, carboxylic group, NH2 group, amino group, hydrogen here, R group here. I have written the general formula of amino acid. This is the amino group. You remember, it's because this becomes basic in nature. The amino group has to be removed. When an amino group is removed, that becomes a keto acid. Keto acid. And that keto acids can enter the respiratory pathway after a process called deamination. Deamination means removal of the amino group. Students, brief idea I will give you. 2 points I should mention to you. In a chapter mineral nutrition, which is we will be studying, where there is a process called as transamination. We will be studying that, where amino group is added to keto acid. But here amino group is removed from the amino acid, then the product is going to be keto acid. That much you remember for time being. So, when the amino group is removed, that process is called as deamination. Please remember, and the product is called a keto acid. I am writing here. So, the amino acid is going to be undergoing deamination process. Deamination process. And during deamination, that amino group is removed. Then you may ask, sir, what happens to that amino group? In the case of humans, if I take it, or other animals also, that amino groups enters to urea cycle and become urea. Ornithine cycle we told the same. Amino group will becoming urea and we excrete as the ureotelic organisms, we are right. Okay. So, the product after deamination is called as what will be keto acid. These keto acids, depending on which carbon number they have. If they have 3 carbon number and the keto acid, if it is a pyruvic acid, then the keto acid, if it is a pyruvic acid, then the keto acid will enter through the pyruvic acid to the respiratory pathway. If a keto acid is not pyruvic acid, but if a keto acid is the acetyl-CoA, then it enters the respiratory pathway through acetyl-CoA. There are so many keto acids comes in the Krebs cycle. So, the keto acid can enter the respiratory pathway through any intermediates of Krebs cycle. Clear, students? That means how many ways this amino acid, how many ways a amino acid can enter the respiratory pathway? If a question asks, answer is 3 ways. One is through the pyruvic acid, which is having 3 carbon, or through the acetyl-CoA, which is having 2 carbon, or any intermediates of Krebs cycle. Krebs cycle can have intermediates having 6 carbon chemical like a citric acid, isocitrate, or it may be alpha ketoglutaric acid having 5 carbon, or it may be succinyl-CoA, succinic acids, fumaric acid, malic acid, or oxaloacetic acid. All these are having 4 carbon. Depending on the carbon number, they enter there. Clear, students? So, that means, to sum up once again, carbohydrate can be a respiratory substrate, fat can be a respiratory substrate, protein can be a respiratory substrate. clear, right? Glucose enter the respiratory pathway, how you know it? We get 38 ATP. A fat can enter the respiratory pathway in two ways, through the glyceraldehyde 3-phosphate or through the acetyl-CoA. A protein can enter the respiratory pathway in three ways. One is through pyruvic acid, second acetyl-CoA, third any intermediates of the Krebs cycle. When the concept of transamination you understand, things will be still better for you. But still you remember, time being that the proteins after becoming amino acid, after undergoing deamination, enter the respiratory pathway depending on the carbon number, means which fatty acid they are. Okay. So you tell me now, till now whatever we discussed, with that you understand that respiration is a catabolic pathway, because everywhere breaking down, because protein has a highest carbon number broken down into less carbon number, ultimately all the carbons are evaporated as a carbon dioxide. It is a catabolic process. I can tell it is a catabolic process. Catabolic process. Catabolic, so I will write a catabolic pathway. Catabolic pathway. Catabolic pathway. Catabolic pathway, catabolic pathway, catabolic pathway. But when, what can happen, I am telling you. This all the molecules, we have seen some of the reversible reactions, right? Reversible reactions. To give an example to you, a pyruvic acid has been formed. After forming pyruvic acids, the cell feels that oh, there are already enough amount of ATP, but amino acids are less. So then pyruvic acid entering into the next step of aerobic respiration, where it has to become acetyl-CoA. Let us not do that. Like a cell tells pyruvic acid, like an imagination, a pyruvic acid, now no need of becoming acetyl-CoA, because ATPs are already there. You have to escape the respiratory pathway and you need to become amino acids, because there is an urgent need of proteins in the body. So in such case, what happens? The pyruvic acid will escape the respiratory pathway and become an amino acid. Then amino acid will become ultimately what? Tell me, proteins. One example. Similarly, pyruvic acid that forms a particular amino acid. Pyruvic acid becomes acetyl-CoA. Then a cell tells acetyl-CoA, no need of becoming, no need of going for further steps of Krebs cycle, you have to become amino acid. So you escape the respiratory pathway, and you please become amino acid, and amino acid becomes later proteins. Similarly, intermediates of the Krebs cycle, I will give one example to you. Alpha ketoglutarate acid having 5 carbon, right? Alpha ketoglutarate acid supposed to become succinyl-CoA. It does not become succinyl-CoA, instead it escapes the respiratory pathway and becomes amino acid. Which amino acid also I can tell you? The amino acid name which is formed by a alpha ketoglutarate acid is a glutamic acid. Glutamic acid is formed by the alpha ketoglutarate acid. So here we saw that they are becoming a large molecule. A smaller molecule is becoming a larger molecule. So I can tell this process as anabolic pathway. Anabolic pathway. Agree with me, students? Anabolic pathway. Let me come to here. Students, the acetyl-CoA can escape the respiratory pathway and can go in this direction to become fatty acid, and fatty acid ultimately becomes fat. Another way, that is regarding fat over. Glyceraldehyde 3-phosphate can also become glycerol and it becomes ultimately fat. So here also I can tell it's a what, tell me not? It is the anabolic pathway. Anabolic pathway. Clear? Catabolic pathway, anabolic pathway. Okay. Now, same thing can happen in the case of the carbohydrates also. Clear, students? Carbohydrates also same thing can happen. That idea also we have, little bit idea also we have it. Okay, fine. So all together if I take it, students, since I will be writing here, okay, I will be writing here, since the respiratory pathway, has or can be better word, can be catabolic, anabolic, because both are possible. See, catabolic, anabolic, catabolic, anabolic, catabolic, anabolic. So it is better to call it as what to me, amphibolic pathway. I am using the word pathway, remember, amphibolic pathway. Respiratory is respiratory pathway. Don't use the word respiration, I am telling pathway. Respiratory pathway can be amphibolic pathway. Clear, right? Is the matter very clear, right? Okay. So this has, so I think I have justified the word how respiration can be a amphibolic pathway. I think things are very clear for you, but I will leave one question to you. Two questions let me leave to you. Two questions. Let us see, can you answer or not? In the muscles, which muscles? The white muscles, skeletal white muscles, during the time of anaerobic condition, there is a formation of lactic acid. This lactic acid has how many carbon? With 3 carbon. This lactic acid undergoes something called Cori cycle. Which cycle? Cori cycle. And that Cori cycle is a what kind of reaction? What kind of reaction? I will give clue to you. Is it a glycogenesis? Is it a gluconeogenesis? Or is it a glycogenolysis? And what is this? Tell me now. The Cori cycle is a what kind of reaction? Is a Cori cycle has something to related to this? First question I asked you. The first question was regarding Cori cycle. Second question, Cori cycle is regarding what? Animals. In the lipid-rich seeds like a castor seeds, rich in fat, they want to germinate. To germinate, they require carbohydrates. Fat is there, but they want carbohydrate. What happens there? Which organelle is involved there? Can you just comment on this? Because you know this. The first question I asked regarding Cori cycle, for that you would refer B07 material chapter locomotion and movement. I am making the things very easy for you. Second question regarding the fat rich lipid rich seeds like a castor seeds, which is going to become carbohydrate, you have to refer a concept of microbodies. Think about this, what kind of reactions? Think and answer, please. Okay. So this has to be remembered. I have some questions for you. You have to think and answer. How many ways a fat enters the respiratory pathway? 2 ways. Glyceraldehyde 3-phosphate, acetyl-CoA. Second, how many ways a protein enters the respiratory pathway? 3 ways. pyruvic acid, acetyl-CoA, any intermediates of Krebs cycle. I have one more question, and what is that? Which is the chemical which is a common for all three substrate? Protein, carbohydrate, fat. For all of them, one intermediate is a common. Who is that? Tell me, who is that? Yes, see, a fat can enter the respiratory pathway through acetyl-CoA. A protein can enter the respiratory pathway to acetyl-CoA. A carbohydrate can enter the respiratory pathway through acetyl-CoA. So, the acetyl-CoA is a common member for all three respiratory pathways. It is a acetyl-CoA. So, please mark it very important. Already appeared question in the competitive exam. Having how many carbon? 2 carbon. Yes. With that, I have once again told you that respiratory pathway is the amphibolic pathway. It can be catabolic, it can be anabolic depending on the need of the body. Yes. That means one assumptions we told, none of the intermediates escape the respiratory pathway. That assumption is assumption, means actual reality is not correct. If you assume that, we get 38 ATP, but that actually not happening in our body. Now, I am heading forward for next aspect. This is what I was discussing, students. Please take down, and you already noted down. Here important one is the glycerol has to be connected to glyceraldehyde 3-phosphate. That correction you have to do it. That connection you have to do it in the textbook. And they have given us a pyruvic acid here. You have to connect it to the acetyl-CoA and intermediates of Krebs cycle. This line you need to draw it, so that it becomes easy for you to study. This you please do it. In a competitive exam, they can erase any of them they can erase. They make an A, B, C, D, and you should be able to tell which are those. CO2, H2O is going outside, CO2 is going out. This also has to be remembered. Now, we will be entering into the next one, and what is that? It is called as respiratory quotient, respiratory quotient or respiratory ratio. What is that? It is a very easy one, friends. You can see that actually. RQ or respiratory quotient, it is given there. Okay. RQ is equal to, it is a volume of CO2 evolved and to the volume of O2, volume of O2 consumed. That ratio is called RQ value. First of all, many times for the student confusion takes place. CO2 above, O2 below, or O2 above, CO2 below. How to rectify it? See here. Among the alphabet C and O, if I take it, which alphabet comes first? Tell me. It is a C. Next comes O. So, C has to come first, O has to come later. So, the C has to come first, O has to come later. Clear, students? So, there is no doubt it is a CO2 which is evolved and O2 which is consumed. Let me explain this with the help of one example. See here, C6H12O6 in the presence of 6O2 becomes 6CO2, 6H2O and lot of energy in the form of ATP. This is regarding aerobic respiration, 38 ATP. Take this equation, students. RQ is equal to CO2 evolved. How many CO2 evolved? 6 CO2 evolved. How many O2 consumed? 6 O2 consumed. 6 divided by 6, answer is, tell me, 1. 1 is the RQ value. 1 is the RQ value for what? 1 is the RQ value for glucose. 1 means complete. So, the RQ value for the glucose is 1. So, the glucose is a favored respiratory substrate. See now, depending RQ value depends upon type of respiratory substrate. That means the question comes, now I have taken carbohydrate, glucose. So, the RQ value become how many? 1. Is the RQ value is going to be different for different substrate? Answer is yes. It's a 1 for carbohydrate. For remaining what? Little later I will tell you. Next one, RQ value for carbohydrates 1 because equal amount of CO2 and O2 are evolved and consumed. So, it becomes how many? 1 RQ value. RQ value for carbohydrate 1 I already told you. RQ value for fat is less than 1. RQ value for protein is a 0.9. These 3 value you have to remember by heart and remember. 1 for carbohydrate, less than 1 for fat and 0.9 for proteins. So, we can tell that for fat as well as proteins the RQ value is less than 1, less than 1. That means you should understand here less than 1 means the O2 requirement is more than the CO2 evolved. Clear, right? Okay. So, I will go for the next one now. The next one example has been given in the textbook regarding one of the fatty acid, and with that several times questions have appeared. Let us take that, and that one is here, students. If you see here, RQ value for fat is less than 1. For a fatty acid called tripalmitin, tripalmitin is one of the fatty acid. Regarding that, explanation has been given in textbook, where 2 molecules of tripalmitin, the tripalmitin is having the formula like C51 H98 O6. See, you know, C51 5 plus 1 6. The way of remembering is a tripalmitin. In the presence of 145 oxygen, it is going to give 102 carbon dioxide plus 98 H2O. See here, this H is 98, H2 also 98. This has to be remembered, plus a lot of energy. That means RQ is equal to CO2 evolved. How many CO2 evolved? Tell me. 102 CO2 evolved and 145 O2 consumed. The RQ value becomes 0.9. So, 0.9 means less than 1 in bracket. Less than 1 is a RQ value for what? Tell me. RQ value for the fatty acid called as tripalmitin. This has to be remembered, and I already told you the RQ value for protein is how much? 0.9. The last word. So, in the living organism, respiratory substances are often more than 1. Means it can be carbohydrate, fats, or it can be even the proteins. Pure proteins and fats are never used as respiratory substrate. Means pure fat is not used. Then what is used? Glycerol can be used, fatty acid can be used. Pure proteins are not used, but what can be used? Amino acid becoming keto acid can be used as respiratory substrate. All those lines has to be underlined and remembered. I am erasing this. It is regarding tripalmitin, which can be used as respiratory substrate and gives RQ value of 0.7. Now, I will take one example of the anaerobic respiration. Anaerobic respiration, just to calculate the RQ value in the alcoholic fermentation, where C6H12O6 plus O2 is not there. So, directly writing the ethanol having, tell me what, 2 ethanols having 2 carbon each plus 2 CO2 and energy. The RQ value here, tell me what? The RQ value here is going to be CO2 evolved. How many CO2 evolved? 2 CO2 evolved. How many O2 consumed? 0. So, what is the answer? The answer is infinity. So, infinity is the RQ value for when the respiratory substrate is going to be what, tell me? It is going to be carbohydrate. Is the matter clear, students? It is regarding the RQ value concept. We had discussed RQ value for the carbohydrate, it is 1. RQ value for the fat, it is less than 1. Best example 0.7 for tripalmitin. RQ value for protein is 0.9. RQ value for anaerobic respiration, ethanolic fermentation is how much, tell me? It is going to be infinity. Clear, right? Can a RQ value be more than 1? Can a RQ value can be more than 1? Answer is yes. It can be more than 1, which will be dealt in the AIC classes when it can be more than 1, but you can 100% search, and if you get the value, if you get the idea regarding that, please share with me, because very easy, the concept is very easy. You can get idea regarding that, where RQ value is going to be more than 1 also possible. If RQ value should be more than 1 means number of CO2 eval should be more compared to the O2 consumed, right or wrong? Yes. Okay. Now, I am going to finally wind up this chapter, after giving a idea to you regarding the entire process in a brief, quickly I will tell you, in brief, fine, fast will tell. Glucose is a favored respiratory substrate which undergo 10 steps of glycolysis, and the 10th step of glycolysis, which gives here 2 ATP net yield and 2 NADH plus H plus. Yes, that is in the glycolysis occurring in the case of cytoplasm, and 10 different enzymes are there, and 1, 3 and 10th were irreversible steps. Product is called as 2-pyruvic acid, means 2 molecules of pyruvic acid, and each pyruvic acid has 2 carbons, 3 carbons. Glucose has 6 carbon. Pyruvic acid enters into the mitochondria. So, pyruvic acid will enter to mitochondria. Yes. So, in the mitochondria, in the presence of what will mean? In the presence of the oxygen. So, it is going to be aerobic respiration, right? It is going to be aerobic respiration, where what happens, tell me? In mitochondria, what happens? The chemical formed where pyruvic acid, who has entered the mitochondria, will become acetyl-CoA. And during this, NADH has been formed. Pyruvic acid has 3 carbon, acetyl-CoA has a 2 carbon, and 2 molecules of pyruvic acid. So, the 2 molecules of acetyl-CoA. The acetyl-CoA later will enter to the Krebs cycle. The Krebs cycle, where H2O has been released and CO2 is also released, complete breakdown it is, and leads to formation of, tell me how much, leads to formation of the 8 NADH plus H plus, 2 FADH2 and 2 molecules of ATP. Clear, right? That ultimately when you calculate this ATP to ATP, this add it here 10 NADH, 10 into 3 30 plus 2 32 plus here 4 ATP 36 plus 2 38 ATPs are formed by the aerobic respiration. Got it right? Okay. But in the absence of oxygen, it undergo anaerobic respiration. And anaerobic respiration, which is occurring in the absence of oxygen, which is also called fermentation. fermentation process is occurring, where tell me it can be two ways, a alcoholic fermentation, can happen where the product which is formed is called as ethanol, having how many carbon, tell me, 2 carbon ethanol, and plus CO2 is formed, decarboxylation is there, and 2 ATPs are formed here. Clear, students? It can be lactic acid fermentation. Lactic acid fermentation can occur where the product is called as what, tell me? The product is called as lactic acid having 3 carbon, plus CO2 is not there, but 2 ATPs are there. So, net yield of ATP is how many? 2. This entire process which I am discussing is also occurring in the case of what, tell me? It is occurring in a cytoplasm. Occurring in a cytoplasm in the anaerobic condition. Clear, students? Number of ATP is less or more? Less. And it is incomplete oxidation. It is incomplete. It's incomplete oxidation, and this is the complete oxidation. Complete oxidation. So net yield was how many ATP? Tell me, 2 ATP. So it was a bird view regarding the entire process which we discussed till now. After consuming lot of hours we had discussed. So all this aspect has to be remembered. Hope everything understood for you. Any doubts, if it is there, please post it in the doubt portal, and keep studying. You have already set the aim for the NEET exam. Do very well. The world is competitive. It's a matter of survival of fittest. I know that you are all fit, but prove it. Let's meet in the next hour in a AIC level 1 discussion. Till then, thank you, students. Take care.